Hi.
Hello, I know you.
Fine. Nice to see you.
Nice to
How
see
is
you. Sorry?
how is how are you?
Yeah, I'm doing fine, thanks. Yeah, you?
Yeah, fine. Yeah. Oh, really good.
Yeah,
Okay.
that's really good.
No, it's bad. Oh, 1 second that I admit. Yes. Okay. I just prepare everything.
Okay.
Good morning.
Good morning.
Good morning.
Okay, here we are. It's 9:30 in the morning, so we can start. Good morning, everyone, and welcome to Neuroccino. Today, we start the month of June with Thalamus, with really an interesting paper on a specific area of the brain. But before to start, did I pronounce your name correctly?
Yeah, very correctly, thanks.
Okay, perfect. Happy to hear. Okay, so you should be able to share your screen. So when you want, you can start.
Okay, so I have the rights to share. Okay, got it.
Yeah.
Can you see my screen?
Yes, perfect.
Okay, wonderful. Good morning, everyone. Good morning from Istanbul. Normally, I'm based in Neuroccino, but for the summer, I'm in Istanbul. Okay, so my name is Talat. I'm a postdoc at Max Planck Institute for Psycholinguistics. You can hear me well, right?
Yes, and maybe can you share the link of the paper?
Yeah, of course.
So in this way also we can have it. Thank you.
All right. First, I need to search.
Yeah.
Okay, there's the DOI number. Okay, I'm gonna share it here in the chat field. I'm trying. Okay, I got it. I don't know, my computer is slow. Sorry. Let me copy it, paste it again. Here we go. So I just shared the link in the chat field. Okay, back to sharing my screen. Not this one. Okay, so you can hear me and you can see my screen, right?
Yes.
Okay. As I was saying, I am a postdoc at Max Planck Institute. And for the time being, I'm in Turkey to collect some data. And today I will be sharing with you a review paper published in Nature Reviews Neuroscience 2023 in May. So it's a very recent article. I wouldn't have noticed it if it weren't for Stephanie, who shared the article with me. And then I said, oh, thank you, I like it. And then she said, why don't you present it? So yeah, and then I said yes, without having read it first. It turns out it's quite detailed, but I'll try to, give you an overview of what I understood and why I chose this paper. Okay, so this is the first time I'm presenting here, so feel free to, cut me and just interrupt me whenever you feel like it. Okay, so the article is titled "The Impact of the Human Thalamus on Brain-Wide Information Processing." Why did I choose this paper? Let me start this way. So I have been interested in the brain, obviously, in regard to language, but I have mostly done things on the cortex, fMRI, looking at the cortex for sentence processing in Chinese as part of my PhD dissertation. But more recently, I have been intrigued by the subcortex because it keeps popping up whenever I look at studies and also in my recent papers, when I looked at connectivity of language areas, most of the time I see this region, especially thalamus, popping up in addition to a bunch of other subcortical regions. That's why I've been interested in this structure. Okay, so then let's dive in. All right. So I don't need to explain anything else beforehand, right? So I'm just starting with the paper at this stage. Okay, wonderful. And I need to check time because I think it's up to 20 minutes. Please let me know if I go beyond that. Okay, so we know that thalamus is highly connected. It's connected to various subcortical as well as cortical structures. And in my, foundational studies of the brain, I always saw this region. I always heard this region as a relay station for sensory motor functions. But I always kind of thought, what is a relay? I mean, what does it do exactly? So this question is a little, I guess, difficult to answer. And most of the time, I think research on this area has focused on lower cognitive functions. Again, sensory motor functions most of the time. Okay, so at this point, maybe I can just show you, instead of the text, I can show you one of the figures. If I scroll down and maybe zoom out a little bit. Okay, here on the left, we see a representation of the thalamus and its connectivity to various structures. This is like a sagittal section. Thalamus in the middle. As you can tell, it is connected to many structures, including brainstem, superior colliculi, the cerebellum, basal ganglia, and the cortex, as well as other areas. And these connections are sometimes excitatory, like the one coming from the cerebellum, and sometimes inhibitory, like the ones more locally located, like the reticular nucleus inhibiting other thalamic nuclei, as well as basal ganglia, again, inhibiting the thalami. Okay, so I think this model illustrates how densely this area is connected. So let me scroll back up. Okay, so this connectivity, this dense connectivity places thalamus in an ideal position to shape multiple aspects of brain dynamics and contribute not just to lower level functions, but also other cognitive and behavioral functions, which could include, for example, language, reading, music, etc. And indeed, actually, so this is no news, right? So there is a lot of research on aphasia, which links thalamic regions, thalamic lesions with phasic symptoms, including, for example, semantic paraphasias. For example, if you want to say apple, if you end up saying orange, then so this is a maybe typical paraphasia example, semantic paraphasia example. So it has been associated with language-related functions. Okay, if you go this way, maybe I just put you guys here to the left. And okay, so so we know that it is connected, highly connected, and we know that it is connected to cortical areas that we are mostly interested in when we do language, for example. So a question is then, why don't we include this in our models, language models, for example, or higher cognitive models in a systematic way? Obviously, there are some attempts, but if you look at Friedrichi, if you look at Hickok and Popple, if you look at other authors, you see that this region doesn't show up in their models. Of course, it shows up in their explanations, maybe in their text, but usually it's not represented very distinctly in language models. And so this paper and a bunch of other papers that I read before highlight some biases, cortical biases in neuroimaging literature. For example, so if you look maybe I just do this. For example, authors have highlighted that we as language researchers, for example, mostly have cortical regions of interest if you want to do post-hoc analyses. So after looking at whole brain, if you don't see activations where we expect them to be it left inferior frontal gyrus or posterior temporal lobe, we have regions of interest which are almost never subcortical. And we have, so we make use of surface-based techniques as well, including MEG, EEG, FNIRS, TMS, which are again mostly, of course, there are some advancements in certain techniques like TMS to include deeper structures, but mostly these are surface-based, and they also increase or aggravate the cortical bias or the corticocentric view. And so subcortical structures are also tiny, small. So this also introduces additional noise or problems when we think about spatial resolution of fMRI, etc. And in case of fMRI, we have hemodynamic response functions which are often optimized for cortex. But when we think about subcortical structures, including the thalamus, maybe their time course of activation, not maybe actually, their time course activation is sort of different from that of the cortex. So we may not be catching the activation that is actually in there when we do classical analytical approaches in fMRI. This paper also mentions ventricular noise because the thalami are close to the lateral ventricles and also maybe other ventricles, so which makes it, again, which introduces additional noise, I guess. But I'm not very, so the paper also says perhaps overemphasized. So I don't know, maybe this is not that big of a problem anymore, I don't know. Okay, so there are also some good news because we are increasing our fMRI techniques to have more resolution, especially with 70, etc. Okay, so here I think I can go to box 2 to highlight a few more things about this corticocentric view and some evidence against it. Okay, here in this box 2, they highlight the past, the present, and the future of neuroimaging the thalamus. Again, so I'm not going to go over the same things, the corticocentric view for higher cognitive functions, including cognitive control, which they take as an example, as a case in point, as well as working memory. So, so these functions, working memory and cognitive control, they have been associated with cortical networks, including frontoparietal systems. But few studies actually focused on thalamic nuclei. Okay, so let me quickly see what okay. Here I also want to make some self-criticism. In my PhD, when I published my first, let's say, series fMRI study on Chinese, I saw activations in a bunch of regions for sentence processing, including amygdala. But although I saw consistent activation in amygdala, it never showed up in my discussion, in my abstract. I just kind of didn't interpret it at all. I ignored it, mainly because I didn't have any explanation for that. So this paper also says sometimes when we find subcortical involvement for higher cognitive functions, we tend to ignore it or we tend to explain it away rather than bringing it into our discussion or interpreting it in more detail. And they carried out a, sorry, I think this isn't them, this is another paper, sorry. So in another study, there was a meta-analysis based on Neuroccino, which is a database of functional magnetic resonance imaging experiments. You can search for key terms like working memory, in this case, working memory and cognitive control, and you can see whether certain brain areas are more activated, more reported for these functions, for these keywords, than would be expected by random association. So based on this, they say that in addition to classical frontoparietal associations, frontoparietal network for working memory and cognitive control, they also saw thalamic activations in specific thalamic nuclei for this meta-analysis for working memory and cognitive control. Okay, so going up. We also check the time. Oops, too much up. All right, so based on this background, they suggest that thalamus is more than lower cognition, and it's a structure which facilitates flexible, multi-scale, coordinated dynamics, characterizing arousal, cognition, and conscious awareness. And they propose a bunch of functions for the thalamus, a repertoire of functions for the thalamus. I think I will highlight these using another figure. Okay. All right, so here is actually a summary of their article. So in this figure 1b, so what they say is, so these are the roles, crucial roles or canonical functions of the thalamus. Activity, coupling, topology, and variability. I can briefly summarize them before I give some more explanations. So activity is mainly modulating, it's about modulating local cortical activity. So, so in this case, we are not talking about networks or different areas. We are talking about the thalamus modulating the activity of a single brain area or a collection of neurons. More details later. Coupling is mediating interregional connectivity. So this is more about not yet networks, but starting to look like networks. And we will say that this is based on neural oscillations, by modulating neural oscillations like alpha, gamma, theta bands across two regions, for example. And thirdly, they also say that thalamus plays a role in cognitive functions by modulating different functional networks. For example, maybe the language network, if there is anything as such, or let's say frontoparietal network, or a salience network, or a multiple domain network, etc. So it's like a hub, a connector hub, so to speak, which modulates these networks. So about variability, so I will probably not talk about this that much, so I'm just gonna skip that for now and go back to the first function, which I intend to talk about a little more than the others. Okay, so what do we have here? Okay, so from mice and non-human primate studies, these are mostly electrophysiological studies, we know that across behavioral domains, including, for example, attention, motor coordination, working memory, sleep-wake cycles, based on these non-human studies, we know that the thalamus is capable of enhancing or inhibiting local activity patterns in cerebral cortex. So this is, again, remember, it's about modulating local cortical activity. Okay, so how does it do this? It's suggested that the thalamus performs this function through inhibitory reticular, by recruiting the inhibitory reticular nucleus. If you remember from the figure down below, the thalamus receives inhibitory stimulation from this area. Indeed, when in, again, animal models, in this case it's monkey, when they pharmacologically or optogenetically, inhibited, optogenetic is, I guess, I heard it before, but I learned it in this article in more detail. It's like inhibiting neurons using light, I guess, by, through light-sensitive neurons, let's say. So when they inhibited the pulvinar or the reticular nucleus of the thalamus, when the monkey performed a visual selective attention task, so this reduced attentional modulation of evoked responses in the visual cortex. So here the idea is the thalamocortical interactions are necessary for attentional amplification of task-related responses. So it modulates the activity of the visual cortex. I guess that's what the take-home message is. Okay. All right, maybe I move you guys to the right again. All right, so there's a lot of literature cited in between, which I'm skipping right now. So most of this research, as I said, is based on electrophysiology, again, which shows that the thalamus modulates cortical activity in humans and also in mostly in animal models. But there's also some more direct causal relationship of the thalamus modulating cortical activity, and we see this here. So in this study, the subjects which had epilepsy, refractory epilepsy, they had deep brain stimulation systems implanted in the thalamus, which could be used to stimulate either the mediodorsal thalamus or the anterior nuclei of the thalamus. So what they found is when they stimulated or inhibited the mediodorsal activity in the thalamus, this affected the subject's working memory performance. But stimulation or inhibition of the anterior nuclei did not result in such a change in performance. Okay, so this is maybe more causal association of the thalamus in modulating cortical activity, especially for a higher functional domain, such as working memory. Okay. See what I want to say here. So they then ask, okay, so for certain functions we have more evidence that, and we know more how thalamus modulates cortical activation, but for many other cognitive functions, including language, we have some models, but still there's a lot to do. We don't know exactly how the thalamus comes in when modulating language-related activity of the cortex. But I might be oversimplifying. I know that there are many models about this. And if you have encountered or if you know more about this, of course, you're welcome to speak up anytime. Okay, so I can admit people. Yep. All right, so this was the first function that they highlighted, activity, modulating local activity, local cortical activity. Okay. All right, so do I have time? Maybe 8 minutes? Okay. I should be quicker. All right, so the second function they highlight is mediating interregional coupling. So as shown here, over here. So let me see what I want to say here. Okay, so how does the thalamus modulate activity of two regions, for example? So or how does it help them transfer information among two regions? They suggest that this may be through coherence of local neural oscillations by modulating different classes of oscillations, alpha, beta, gamma, etc. oscillations. So they have this linkage between the thalamus and these cortical sites. So they say that these oscillations are enabled by GABAergic inhibitory neurons linking cortical sites, which these inhibitory neurons, which are themselves linked to thalamocortical projections. So they suggest this may be a mechanism for the thalamus to couple brain cortical sites. Okay, let's see. And this in turn can facilitate information transfer between cortical regions. For example, as has been suggested, between left inferior frontal gyrus and posterior temporal lobe for more integration or maybe, I don't want to be too strong, but more grammatical or syntactic functions for language or unification functions for language. Okay, going down. I guess I'm not going to say much about this. Oh, no, actually, this is, let me see. Sorry. There's a lot here. Okay, so yeah, let's say it's enough for this one. Time is running. Topology and dimension. How much time do I have? Sorry.
Well, now we are close to the end, so maybe if you can
okay.
sum up a bit,
Right.
and then so in this
Right.
time we have a bit of time for discussion.
Okay, wonderful. Then I will just quickly summarize this one and then finish. So topology is about the thalamus being a connector hub. The thalamus being a connector hub across multiple networks, so serving integrative functions. So there is a lot about graph theory here. I'm going to skip that. It's known to be suggested to be a connector hub because it pops up in many functional networks, as I said before, including frontoparietal networks. Let me see. Okay, and there was some good stuff over here. Sorry. Okay, then if it serves this integrative function across functional networks, then we should expect widespread effects when the thalamus is compromised, right? So this is a prediction of this connector hub view. Do we see that? Yes. So thalamus lesions are associated with impairments across multiple domains, usually, which include language, memory, executive function, and attention. So there is also some evidence to support this connector hub view, which I guess finalizes this little presentation. And so I chose this. I actually explained it already. So I chose it because I'm interested in the thalamus. I'm interested in brain networks rather recently, so I might have oversimplified certain things. Forgive me if I did, and looking for your feedback. Yeah.
Okay. Thank you very much for your presentation. The paper is really interesting and the message is clear. So we should consider and analyze this area more. But now, fellow participants, do you have any questions? Yes, I see a hand. Okay.
Yeah, thank you for the presentation. About the different functions like topology, coupling, activity, do they discuss if they are performed by the entire thalamus or by different nodes, or is the division of the thalamus more in terms of connections with different regions?
Yeah, a very good question. Thanks, Chris. Indeed, they actually, so they say we should discuss this whenever we, see thalamus activation. It's not just thalamus. We should focus on which particular area is involved, might be involved in different function. And they have they summarize some literature. I don't know if I can quickly go there. It's under topology. So they say, for example, let's see. So I in my last few sentences, I said it serves like an integrative function across multiple functional domains. They say that, come over here. Okay, they say that, for example, lesions to thalamic regions with weaker hub properties have a more limited impact on behavior. So they say the thalamus, it has multiple nuclei. For certain nuclei, like mediodorsal nucleus, it's more, it has been shown to be involved in multiple functional domains, and it's, what do they call it? Sorry, its participation quotient or participation number in these networks is higher than some other nuclei. And indeed, when there is damage to this particular nucleus compared to others, there's more widespread functional impairments. So indeed, there is this sort of gradient in the thalamus, as they say, and certain parts of it are serve more integrative functions than others. But beyond this, I don't know. I think it's still a lot. There's still a lot to do in that regard. I mean, which
also with
part of the
MRI, it's
thalamus?
difficult to capture
Exactly.
the different nodes of the thalamus, right?
Yeah, exactly. Maybe we need to directly measure from that area, right? I mean, so instead of the whole brain, sometimes you can also do that, right? You can specify your analysis area. Even with 3 Tesla, you can still get quite a good distinction across these nuclei if you specifically focus on the thalamus, which we don't do usually. So,
yeah.
Yeah.
Thank you.
Yeah. Thanks.
More questions? Otherwise, it's already 10 o'clock.
Yeah.
So yes, thank you very much again for your presentation, and thank you to everyone for attending. So have an exciting science week and see you at the next Neuroccino with another interesting paper to know.
Yeah, thanks a lot for this opportunity. Yeah, good to see your faces. Yeah, bye.
Yeah, bye-bye.
Bye.